Thank you for joining us, and I apologize for the delay in starting the lecture. It is really truly our honor to receive Professor Jean-Jacques Muyembe-Tamfum and the team from INRB, as well as Mérieux Foundation Labs at the Needle. And in particular, what an incredibly opportune time for you to be here, as we are struggling in the second of the emerging infectious diseases threats in this country. Just for the audience who are listening, I'm going to introduce our guests, and then I'm going to pass the baton over to Dr. John Connor to introduce their panel and go through the questions and discussions. And I really encourage everybody in the audience to take advantage of sort of the intellectual capital that we have here today. So just to start, Dr. Muyembe-Tamfum is the General Director of INRB and has served in this role since 1998. In addition, he currently serves as a scientific advisor to the WHO and as a professor of medical microbiology and virology, Faculty of Medicine at the University of Kinshasa, DRC. Dr. Muyembe is known for his pioneering work on Ebola, including co-discovering the Ebola virus, being issued a U.S. patent on neutralizing antibodies to the Ebola virus glycoprotein and their use. He received his MD at the University of Louvain, a Democratic Republic of Congo, and a PhD at the Rega Institute for Medical Research in Catholic University of Louvain in Belgium. We also have with us Dr. Placide Mbala, who serves as the chief of the Pathogen Genomics Laboratory at INRB. Just incredible experience, in the past with working with emerging infectious diseases pathogens, and we were just speaking about the recent Ebola outbreaks, multiple Ebola outbreaks that DRC's had. Dr. Daniel Mukadi, who serves as the director of Rodolphe Mérieux Laboratory of Goma and is a provincial director of INRB, and brings incredible experience with prior pathogen responses to polio, to Ebola, of course, and Monkeypox, and multiple Ebola outbreaks, I was going to say. And Dr. Hugo Kavungo, who serves as the deputy director of the Rodolphe Mérieux Laboratory of Goma, DRC. Again, big integral part between public health response and research in DRC. And to lead off our panel, I'd like to introduce Dr. John Connor, who you all know is faculty at the Needle, as well as the Center for Emerging Infectious Diseases Policy and Research. Dr. Connor is an associate professor of microbiology, and his work focuses on emerging infectious diseases, diagnostics, and host biomarkers. John, I should probably introduce myself. I'm Dr. Nahid Bedalia. I'm one of the faculty at Needle, and I'm the director of CEID. Thank you.
Thank you, Nahid. First, I want to add my welcome. Thank you guys very much. I understand that you have moved in multiple places around the country so far. It's very nice that you have taken the time to be with us this morning. I thought, Dr. Muyembe, we might start with you, if we may. You have seen the evolution of the INRB and the overall response to these emerging pathogens over a long breadth of time. I wonder if you would comment on what it was at the beginning and how it is now and how you see it's expanded.
Okay. Yeah, thank you for your presence, and I'm happy to see my old friend in front of me. I don't remember the year.
1987.
Yeah.
So old is right.
Yes. I am the veteran for emerging infectious diseases in Africa. So the first year I encountered what we are calling now Ebola virus was in 1976 in a Catholic mission in the equatorial forest, Yambuku. And on 21 September, the Minister of Health called me. I am just returning from Belgium, so she sent me to go and investigate deadly disease. He thought to be typhoid fever or yellow fever. So I traveled with an epidemiologist. I am virologist with an epidemiologist. And when we arrived at the night at this mission, we went directly to the hospital, and the hospital was empty. Only one child in the arm of his mother, and this child will die in the night. So we spent the night. In the morning, the sisters told me that three healthcare workers died in the night. So in my head, I say that may be yellow fever. So we went there and we collected liver fragment with a big needle. Mm-hmm. Yes, and you can imagine blood everywhere. Yeah, and after that, when the population heard that doctors came from Kinshasa with medicine, they started to come back to the hospital. So the hospital was again full of patients. So after a short physical examination, I found nothing particular, and I thought it was yellow fever, typhoid fever, because fever, headache, fatigue, extreme fatigue. So, like a microbiologist, I said, no, I have to determine the etiology of this fever. So, I selected 5 or 3 or 6 patients with fever. I took the blood for blood culture, and also I said it was important also to make a without test. Without test, it is serological test for typhoid fever. But when I removed the needle, the puncture place site continued to bleeding. So it was for me the first time to see this phenomenon. When I was a medical student, I'd never seen that. And at noon, we come back to take lunch with sisters, Belgian sisters. And one of the sisters who was in charge of treatment of patients told me that she had developed fever. So for me, it was critical. So I decided to shorten my stay there and come back to Kinshasa with the sister to find exactly what was going on. So I traveled with the sister side to side from Mumbai, I don't know, you know, and also my clinical samples as hand luggage. So from there, we traveled to Kisangani, and we changed the plane. We took another plane from Kisangani to Kinshasa. I think you can see the map of DRC. And so we arrived at Kinshasa, and sister went to a clinic Galema, where she was hospitalized and treated as typhoid fever by a Belgian doctor who was there. And the next day, I bring my samples to my lab. It is a simple lab like this, with wooden bench covered with formica. And at that time, mouth pipetting was Yes. And so my assistant, my technician, starts to make the reaction, not repeating, and so on. And but the results were not conclusive because the blood culture were negative, and some of the without test were more or less positive. So it is difficult to conclude. And the pathology were also processed at the university lab, and also the conclusion was that 2 samples were compatible with the congestion, liver congestion, and 1 sample were maybe yellow fever. So some day later, the sister died and contaminated 1 Congolese healthcare worker, and the other sister who accompanied this sister. So there was panic in Kinshasa, and the sample from the sister sent to ITM in Belgium, and by electronic microscope, they found a virus that has the same morphology than Marburg virus. So they conclude that it was a Marburg virus outbreak. But when the sample arrived at the CDC, neutralization test and so on, Dr. Carl Johnson concluded that it was not a Marburg virus, but a new strain of virus called Ebola, because there was a small river near Yambuku Mission. Give the name of this river. So this was the beginning of the disease, and 19 years later, for the first time, a urban outbreak occurred in Kikwit, and the symptom was bloody diarrhea. And the bishop there, who was my friend, called me saying that we are losing people here, even the Belgian sister, no, what Italian sister. So if we receive this kind of news, it is exactly a concern. So I traveled there. As in Yambuku, I tried also to find the etiology of this bloody diarrhea, who was not treatable with antibiotics and anti-malaria, and with high case fatality rate. So my bacteriological investigation were all negative. So I thought it is not possible. So I started a small epidemiological investigation to see who, where, and when the disease started. And we conclude that this bloody diarrhea started with a lab technician who processed sample for malaria or for typhoid fever and was infected and came to the hospital. So this kind of disease, where the contamination occur in the operating room, is not dysentery, malaria, bacterial infection. It may be viral infection and hemorrhagic infection. So I collect blood and send to ITM, and ITM sent this blood to CDC, and the conclusion was that it was Ebola. But it was difficult to believe that it's Ebola, because Ebola occurred for the first time in the forest, where people are eating monkeys and so on. And this time it is in the south, more or less 1,000 kilometers apart. And in Kikwit, it is savanna, and we have no monkeys, or what, eaten and finished. So in this region, we had no monkeys. So it was very difficult for the population to accept that it was really Ebola virus. And during this outbreak, my team, at the end, more or less at the tail of the outbreak, we treated 8 patients with the blood of survivors of Ebola, and only 1 died. So for us, antibodies can protect, but our partners, most of them were foreigners, did not believe that, and saying that our experience, the sample were too small, and also maybe because it is at the tail of the outbreak, so the virus has lost its virulence, and so on. So they refused to accept the result. Yes, it is why after during the future outbreaks, we didn't do this, repeat this experience. But in 2004, Dr. Barney of NIH came to visit me at INRB, and we discussed about this experience, and he was interested. And we decided to send some of our survivors of Kikwit to NIH to explore their immunity system. And also, Dr. Nancy of Biodefense accept to work with my team. So 2 of my team went there to work on the blood of Kikwit survivors. And one of them give a very good result. We developed the antibodies who are very, very active in macaque. And they conduct also the first phase of in human. And when the outbreak, the 10th outbreak of Ebola, was documented in Mandina in 2018. So we decided to use these monoclonal antibodies we call 114, MAP 114. After obtaining authorization of ethical committee and also regulatory authorities. So we compare the efficacy of MAP, Remdesivir, Regeneron, and ZMAP, and we found that MAP can protect, is effective when we give to patients 34 of we can cure 34 of the mortality.
Yes, but it's reduced to about 30%.
Yes.
From what? From 67?
From 67.
From 64. Yes. So, and after that, our molecule was accepted, approved by FDA. It is why we are saying that now Ebola is a treatable disease. And also during this outbreak, we evaluated also the efficacy of Merck vaccine, the RVSV is above, yes, that we give ring vaccination. bring vaccination around the case of Ebola. So this outbreak was very important for us because we obtained two most results. Is the vaccine is active, and also we have two monoclonal antibodies to treat the patients with Ebola. So for me, it is my dream that this Ebola was a deadly disease and a terrific disease. We can now treat Ebola. We can also prevent Ebola. So thank you very much.
I would like just to mention
Please.
that normally the treatment with the monoclonal antibody, when the patient have like high viral loads, so less than 22, when the CT value is less than 22, yes, the monoclonal antibody can reduce the mortality to 17 to 30%. But when the viral load is low, so with the CT value over 22, 30, that can be reduced even to 10%. And we also, because we conducted a clinical trial during the 10th Ebola outbreak to show that the two monoclonal antibodies, so our monoclonal antibody, the MAG114, and also the Regeneron monoclonal antibody, both of them were I can say efficient to treat Ebola. And then we used them during the 11th outbreak to treat 32 patients, and the mortality was just 2 patients among the 32 that we treated with both. So you can also find this paper in New England. We publish the paper of the treatment, the monoclonal antibody treatment, 32 patients.
Yeah, and right, I don't know that I can emphasize enough that right, this is a remarkable story of essentially changing how an infectious disease is viewed, right? Where it has gone from something where there is very high mortality and no good answer to where there are now multiple answers to it. I'm very curious about all of your experience with how you think about outbreaks now versus how the outbreaks affected the DRC prior to the development of these tools.
Yeah, I think the problem of Ebola is the capacity to early detection of the disease. If the disease is detected early and rapidly confirmed, so with isolation and the arrival of innovative technologies, so we can control this outbreak quickly. And if I remember, the last outbreak occurred in 2000.
The current one, there is an outbreak right now.
There's a, but it will be quickly controlled. I think this is a problem. And also, to be effective, the treatment, the patient must come early. But most of the time, the disease is ramping in the community because community population thinks that it is malaria or typhoid fever. So they lose a precious time before coming to the hospital and receiving monoclonal antibody. But if this period is short, the chance of of surviving is very high.
So this also brings up, I think, brings up the idea of the importance is shifting, not shifting, but it's more obvious that having a good infrastructure to identify is becoming increasingly useful and important.
Exactly.
And can you, and I think again this is for everybody, can you speak to how the DRC has addressed it and is addressing it?
Yeah, I think Mukadi you can answer. Concerning also our infrastructure in Goma.
Okay, thank you very much. And so before we have those innovative technologies during outbreaks, so most of our response were based only on diagnosis capacity on site. But with innovative technologies, we are ready at the start of the outbreak to deploy the diagnostic capacity, the sequencing capacity. Placide will emphasize on this. And we also deploy vaccines capacity. And for example, Hugo was able to deploy GMG vaccine to prevent Ebola disease occurring from one city, the biggest city of North Kivu. And so those tools are now associated so that in our response deployment and preparedness, we did not only include diagnosis, but we have sequencing, we have monoclonal antibodies, we have vaccines, and we have on-site sequencing capacity. And during Eastern DRC outbreak, which was the longest and second largest outbreak of the world, we were able to have like 13 field laboratories run at a time. And we had a sequencing capacity so that we could give a real-time update and guidance of surveillance, because we were in the context of insecurity, so that we had many unknown chains and some other cases which couldn't be identified. But with the pathogen sequencing on site, we were able to reorient diagnosis, and that triggered the vaccination of contacts of frontline healthcare workers. And we're also able to follow up those patients, because once you administer either molecules, monoclonal antibodies or antivirals, it's very important to organize the follow-up, the biochemistry and the biological follow-up. And actually in our system, we also have those additional tools to deploy in the field for chemistry, for hematology. And we did also add the follow-up or EVD survivors during the outbreak, because during the Western outbreak in West Africa, the survivor follow-up started a little bit earlier, and we had many reintroductions, so many relapses and flare-up of outbreaks in the region. But with our system, the follow-up of EVD survivors starts just with the outbreak, so we are sure that we can prevent from relapses and some other reintroduction. And so this is something very, very important that we use. And from that capacity, we create field laboratories, first for Ebola. And when the Ebola outbreak, for example, ended in 2020, the COVID-19 pandemic has already started. And so we shift our field laboratories for Ebola diagnosis to hard COVID capacity in site. And so far, we still have those laboratories. They have become INRB laboratories, now supported by the Rodolphe Mérieux lab, which is a BCL2, BCL3 facility, as a regional laboratory supporting the provincial response, etc. And something also I'd like to emphasize is the community engagement. Before August 2019, before Professor Muyembe took the leadership of the outbreak, we had all those countermeasures available, but we didn't succeed because we didn't get that community engagement. When Professor Muyembe was appointed, first, he didn't go to the field to talk with medical doctors. He first led community leaders, social traditional leaders, to have the discussion, to explain them what is the disease, how it spread, and how it's very important to have them involved. And once that the step was done, and then we were able to succeed with our countermeasures. So that brings up the necessity of having also a psychosocial component in the response on the top of countermeasures we need to have. Thank you.
I think we have to mention that in the field we have used the existing technology with gene expert. Gene expert is used by tuberculosis TB control. And also for HIV. So now we have the cartridge specifically for Ebola. So as gene expert is everywhere in the country, we can use this on the site of Ebola outbreak. So this is fantastic. And also the same gene expert is used for COVID-19.
And then maybe also something that we Placide, you are working on to have those gene expert cartridges for monkeypox, I think?
Yeah, for monkeypox.
Yes, we are working with the CDC to validate gene expert cartridges for monkeypox. So we think that this will happen very soon. This will change the way to diagnose monkeypox, since monkeypox in our country is also endemic and it occurs mostly in remote areas. So using a gene expert's capacity, as it's already deployed for TB, HIV, and now we are using most of them for COVID-19 also. So if we add monkeypox cartridges, I think it will be very important to also improve the diagnostic of monkeypox in the country. So what I want to maybe emphasize to what Dr. Daniel said, I think the key point in every outbreak, this is what I learned from my modest experience, is first of all community engagement. If you don't get along with the people, if they don't know what is it, if they don't know what you are doing, you will never succeed. Even if you bring the newest, the technology that you have, you will never succeed. So it's the main point. And I remember when I was doing my master in public health, I had this kind of scenario, how to explain to someone who is not the medical science, how to explain him the situation. And I remember that I failed. Because I was speaking like a scientific, with scientific terms, scientific words. They don't understand that. So we have scientific, and I see most of the time a big gap or distance between the scientific and the people. So as long as they don't understand what we do, they will never commit to what we like to do. So it's something that we need to work on it.
So I would like to add also that, as explained Professor Muyembe, it's a long way for research to reach today what we are. So another step which is very important is the preparedness for the whole country. So that, as you know, our country, the DRC, has mostly, we had 14 Ebola outbreaks up to date. So if in each country we did not train the medical people, we expose them to contact these diseases. So the preparedness is very, very also important to contain as soon as possible the Ebola outbreak.
Right, but essentially speaking to the multiple levels, right? There's technology that is helpful,
Exactly.
but there's infrastructure in people, there's infrastructure in knowledge, and in buy-in. right, from the community. I think they're great points. Jerry, did you have a question?
Yeah, I've got a comment and a question. So on the issue of the community engagement, you all know Cliff Lane. And I thought it was absolutely remarkable during the West Africa outbreak, when they finally were able to get clinical trials going. Cliff Lane said, who would have thought social engagement, community engagement was so important? And it's absolutely critical to get the buy-in. Prof. Muyembe, could you think back again to 1976? There you are, fresh out of your training. You're sent up to address this outbreak. You have no idea that there's Ebola. You're handling blood. From your description, you're bathed in patient blood when you do the biopsy and take samples. You sit with a patient traveling all the way back, and you didn't get sick. So I want to know two things. One is, first, I want your immune system. And the second is that you must have had remarkably good training in your laboratory technique to be able to do all of that without knowing that that's the problem and remain healthy. And there's an anecdote that when the samples came to Peter Piot in a styrofoam container, he opened it up. One of the tubes had broken and there was blood around inside the container, and Peter didn't get infected either. So I want his immune system as well.
No, at ITM they have gloves, but in my country we have no gloves.
Then I want your immune system. So as you think back now, why were you not? You must have been exposed. Why did you remain obviously healthy for these many years?
That's a chance for me. I think in Yambuku, when I collected, when I took blood, my hands was soiled with blood. What saved me is immediately I asked to wash my hand with soap and so on and so on. I think this is a very important moment. It was a very important moment for me to wash hands. And washing hands is very important. It can protect you against cholera, against typhoid fever, against COVID. Okay, so water in the community is very, very important. And I test my blood, I have no antibodies for that. But in Kikwit, I was already aware. When you are working on the field of outbreak, you must work with your heart to communicate with the community, and you must work with your scientific experience to avoid contamination. I think these two things are very important in the field. And my technician, I don't remember exactly what happened, but we were exposed in this lab because we use a glass tubes. So we have to wash these glass tubes and reuse these glass tubes. So this is a very important moment, dangerous also. And mouth pipetting is also very dangerous. Maybe we respect the fundamental universal precautions. So this is very important in the lab.
I've asked questions. If people have questions, do raise their hand. I'm happy to call on you. Jackie, please.
Hi, I'm Jackie. I'm a PhD student in John's lab. And so my question was essentially, you guys mentioned that during the 2020 Ebola outbreak, you built up all this community communication, the genomics support and everything, and then you were able to transition up to COVID. and responding to the COVID-19 pandemic. And I just wondered, essentially, how did you do that? Because I think that's a pretty unique experience where you were building up this infrastructure and then had to switch it to a different disease, but were able to maintain it. So what did that look like?
Yeah, and I think our story, we can go back to 2017. Most of the time when there is an Ebola outbreak, we used to wait for colleagues from Canada, from Europe, to bring their expertise, so the lab reagent, and then go to the field, work with them for diagnostic. And in 2018, as we have already learned, in 2012, 2014, 2018, so in 2017. So in 2018, we decided to take over. So to be able to do it by our own, it was very challenging because doing an Ebola outbreak is not only doing the diagnostic, but you have to do the diagnostic fast and right, and then be able to communicate with the AP team. And then you have to, because they are waiting for you to tell them if this case is confirmed or not. And sometimes it's difficult because we were using a kind of, it's an open PCR. And as you know, the Ebola virus has several genes. So sometimes we have primers for like 2 genes, like it could be GP and P. The other primer could be like maybe VP40, VP35. So you have different type of primers. So sometimes you can test the same samples. If one primer is positive, so you get the positive only for NB, for example, not GP. For the other one is negative. So what to tell exactly? See? So they are waiting for you. You have to tell them, is it positive or negative? Because they have to move the patient from the suspect area to the confirmed area.
Yeah.
So it was really challenging. You get pressure, and sometimes we bring the centrifuge in the field. It doesn't work. So we have to wait for another centrifuge. I needed to take the plane, take the helicopter, and go to the field. I didn't know where to sleep. I work in the lab, and then at the night, I don't know where to sleep. I just sleep on the bench in the lab. Yeah, it is very, very stressful because you are getting pressure from WHO, from MSF, from ALIMA, from everyone. They are calling. Yeah, and so I think this helped us to grow a little bit. And then during the Ebola outbreak in North Kivu, we then decided to take over everything. And fortunately, we had also the gene experts. So we deployed like 11, 11 sites. So we collected during that, we analyzed during that outbreak almost over 200,000 samples by our work. And it was also during the same period we had this collaboration with the USAMRI, so they trained us on sequencing. So we are able to deploy a local team in the field to do genomic sequencing. So as the site was very difficult because of armed conflict, unrest situation, so the genomic surveillance helps a lot the APT to, for example, make the link between confirmed cases, to elucidate some cases of relapse, reinfection, and so on. So this is how we go. And then I think due to this experience and with the donation that we received from Mérieux, we decided to set the Goma lab at the North Kivu to make sure that we could be very quickly respond to outbreak that can occur at the eastern part of the country from the Goma lab. Because if you see the map of DRC, you see that traveling from Kinshasa to Goma is a long way. So if we want to ship every time samples from Goma to Kinshasa, it's very difficult. So having another lab in Goma was very beneficial for our team. So we trained more people there, and we have now a new lab in Goma. And then when the pandemic, the COVID pandemic occurred, it was easy because we used the Gene Explorer cartridges also. We had already a good experience on PCR. We had already a good experience on genomic surveillance, so we just applied what we learned to also contain the COVID-19.
Maybe just to add on the top of what Dr. Placide is saying, that first of all in the field, we empowered local staff, because all those labs are run by local staff, and we are just playing the role of supervision. And so it was very easy to switch from one platform to another, since they are the same people used to train, and we kept the same staff as of today in all different laboratories in the field. So that also helped a lot in the way of empowering people. Thank you.
So beside of that, I would like to say that I've seen now the power of communication. Even for COVID outbreak, there was a big problem in Africa to get vaccine because of communication. And if you don't communicate enough with people, they don't agree to get medicine or vaccine. And it was a case of Ebola outbreak. So it was needed to get approval of the local authorities, as said my friend, my colleague Daniel. This is very important to avoid any resistance within the community.
Sorry, I have a follow-up question. When you guys talked about great training, that the big part of that was training locals to stay on site and be able to help with multiple outbreaks. Did you find that also helped with the community engagement portion, component of helping the community engage in treatment and response and participation in contact tracing and stuff like that?
Yes, definitely. For example, contact tracing is done by the surveillance. The surveillance is the outreach team. So we have, for example, someone coming from the central level from Kinshasa, but the rest of the team are people from the community. This is the first. And in the DRCL system,
this is very important because they will speak the language of the population. So the communication is easier.
It's easier.
Yes, and we have what we call community relay. Community relay are not healthcare workers, but people who are living in the community, in the village. So they have the voice to be heard and to talk to people. And our key message are driven by those people. First, before we make message, we do contact them, discuss with them to figure out how is the good way of communicating regarding this disease, how we can say this, and they help us to have the message. Regarding what we are saying, and with NIH clinical trial, we have the SMC team, social mobilization communication team, that goes before laboratory team, and we used to keep that. And also during the vaccination with GNG vaccine, we had a very nice, very good communication team which was on the field in suburbs and quarters to try to explain people what we are doing this activity. It's very important, and also to have the commitment of local authorities. That is also very important.
I think what is also important is to always have specialists of communication working with the local people. It's really important. Why? Because they have to define together which message should be communicated to the population. And it has to be clear. Because if it's not clear, the message will change from one community worker to another. And then when, after 10 community workers, when you get back the message, it's totally different from what you say. So it's important also to have like good people communication and make clear the message that you like to give to the community.
Thank you.
Hi, so what percent of population is vaccinated against Ebola? Is it like required, or do you just do it when there's an outbreak?
Yes, I can say that during the Ebola outbreak, we have something like a ring vaccination with Merck vaccine. So that means the contact of the person who is sick. But apart from that, we had a clinical trial with J&J vaccine in North Kivu, where we vaccinated around more than 20,000 people. This was a clinical trial with J&J vaccine, phase 3. But now we are thinking to go to another province where we had more than 4 Ebola outbreak to get them a preventive vaccine, which is also changing. So we'll start very soon to vaccinate people from that region.
Just to add that during the DRC Eastern outbreak, with Professor Muyembe and Anne-Maria from WHO, we have vaccinated 300,000 people for that outbreak, which lasted almost 2 years. So we included contact for contact, and frontline health workers. That's very important.
So for the moment, it's difficult to say exactly how many people have been, I can say, vaccinated, because it's not a routine vaccination. It's a vaccination that responds to an outbreak usually. So we deploy the vaccine when there is an outbreak to try to control the outbreak around the contaminated cases. So when there is a case, we vaccinate all contact and contact of contact to make sure we are making a ring of security around the contaminated cases. So it's a bit difficult to estimate, because right now there are also, as we are seeing some cases of relapse, some cases of vaccine breakthrough. So we are also now providing like booster to some people who were vaccinated like 2 years ago. Yeah. So for the moment, it's still like kind of one tool to respond to the outbreak.
And also, it's very important to know that we have 2 kinds of vaccines. The first one, the Merck vaccine, is a vaccine to stop the outbreak. It can be only used during an outbreak to stop transmission, human-to-human transmission. But the J&J vaccine is a preventive vaccine, so that can be used before an outbreak occurs in the community, because it's 2-shot doses, and the RVSV, it's a 1-shot vaccine. And the question arising now with WHO and SAGE is to know if we should revaccinate people. And this is what we did for the Ebola outbreak last year in Butembo and Beni, Eastern DRC, and from the ongoing outbreak. Even people who were vaccinated 2 years and 1 year ago were revaccinated, because we are not sure of the everlasting immune system with those outbreaks. So this should be maybe the perspective for next studies to look at the immune response amongst people who were vaccinated for 2, 3, 4 years to see how the immunity is going on. Thank you.
So it seems that some people do not respond to monoclonal antibodies. I think a study was mentioned where 2 out of 22 people did not respond. So I was just wondering if these non-responders are there because they were already past that time window when the monoclonal antibodies can work, or if there could be some strains circulating in the country that could be resistant to these available monoclonal antibodies.
So what I can say is that When Prof. Muyembe talked, he mentioned that there still be a certain percentage of mortality, even when they get monoclonal antibody. When the viral load is very high, it could be about 30% who could die. And when the viral load is low, it could be about 10%. So there are still people, as you know, according to our immune system, everyone can respond differently to a drug. So we still don't know exactly. This is also, I think, a big question to explore how some people do not respond to this monoclonal antibody. But in the term of genomics, we are doing a kind of investigation of mutations at the target site of the two monoclonal antibodies. And till now, we did not find mutation that can impact the efficacy of those monoclonal antibodies. So, but as I said, every time when we are trying to find a solution, those solutions are bringing new questions. So we need to explore and understand why. The first thing, I think, the first thing that we would like to do is to see if increasing the doses of monoclonal antibody can maybe help. So this is something that we are planning to do if there is a new outbreak, setting a kind of maybe two-arm randomized controlled trial where we can give the normal dose and maybe a higher dose to another arm and see if we may get a good response. Another study that we are doing with Daniel also is to see the difference of persistence of the virus among those people who receive those monoclonal antibodies and also people who receive the antiviral, for example. Because we are treating, but we are seeing also more cases of relapse. So it's important to know if it's because of the treatment or it's just the body which is responding to the aggression that is getting to it. So there are more questions that are raised and we need to explore.
Maybe one additional question should be, because on all these outbreaks, it was only the Ebola strains or Zaire strains. But what will happen if we have a Bundibugyo, a Sudan outbreak in DRC or elsewhere in the world? So that will change the paradigm, because we will need additional gene expert cartridges, maybe with different targets. We maybe need other vaccines because the targets maybe look a little bit different. So these are the main key questions we should have in terms of response to viral hemorrhagic fevers to prevent maybe some other outbreaks. Maybe other future outbreaks will be made of Sudan infection, but what will happen? Will be vaccinated with Merck and G&G? We don't know. So it's also very important to have that in mind.
So I'm sensitive to time. We've gone a little over what we said we would. We got started a little late. So I think we'll stop here for the moment. But all of you, thank you very much, Professor Muyembe.